This is feedback for core component mock, and this is mock 2. First question. The diagram shows a structure of a protein, and letters A to C indicate 3 types of bonds. And state the name of the type of bond found in the primary structure. So this primary structure is the polypeptide chain, and it's asking between each amino acid residue in the chain there's a bond between those, and that's your kind of first type of bonds in proteins. That's peptide bond, and it's not a polypeptide bond or a dipeptide bond, so just a straight peptide bond to the primary structure. Name the types of bonds A to C. A, we have between an O and a H on a C2O, and this is electronegativity, so the O takes part of the kind of electron cloud, so part of the minus away from that carbon, and the H loses that to the nitrogen. So these are a little bit, so delta negative, delta positive, and so the type of bond between the little negative and positive is hydrogen bonds. So that's a hydrogen, and the same there. We've got little delta negative. Oh, sorry, that'll be the negative there. And on that H would be delta plus, so hydrogen bond again. And B is here, we've got 2 sulfurs, so that should lead you to the right type of disulfide bonding, or disulfide bridge. And we spell it this new way now with the F. So these are in the tertiary structure, the types of tertiary structure bonding between R groups. So we've got these sticking out here, these are R groups attached to the amino acids, so these are all R groups, there we are. And C, this isn't the delta small positive and negative, it's an ionic charge. So when you see pluses and minuses like that, it's ionic, and so ionic charges interact, and those are ionic bonds. A bit like in salt, sodium chloride, things like that. So you should know your names of the bonds. The area marked X forms a secondary structure, give the name, and so it's trying to show that it's a kind of spiral or spring shape, and you should recognize that as a helix, and the type of helix is kind of left-handed spiral, so alpha helix. So alpha helix, or you might have put the symbol for alpha. How is this form of secondary structure held together? So it's asking what bonds have we got between these, holding these together. Well, these bonds, if I hadn't actually had just scribbled over the top of it, they're like bond A, but these are actually not between the R groups. They're actually the bits sticking out on these peptide bonds. But the answer is the same type of bond as A, which is hydrogen bonds. There's a lot of space to write about hydrogen bonds there, but essentially that's what it is. So hydrogen bonds between the C double bond O and HN in the actual peptide links. State the name of a further form of secondary structure. So if we've had kind of alpha helix, there's the kind of other one, the kind of with pleats, beta pleated sheet. and state the highest level of protein structure shown in the diagram. So we've got primary structure, which is this polypeptide chain. We've got secondary structures, which are the alpha helix sections there. But then we've got further bending and folding of this polypeptide chain, and that's the definition for a tertiary structure. So tertiary structure. And then we go completely off track here and start talking about cellulose. So completely different. Large number of monosaccharide units, and cellulose, it's made out of glucose, but it wants the type of glucose, and 2 marks there. So we're going to have beta glucose, or you might have put the kind of big B like that, and those are the 2 marks. Describe how 2 monosaccharides are joined together. Well, if we just draw beta glucose, so let's draw a carbon chain. So 6 carbons. The ring is completed by oxygen. Each carbon has got 4 bonds. And on these we've got some OHs. So alpha glucose is down, down, up, down. So, and beta is different on the first carbon. So this is up and then down. Got one up there somewhere, and the rest of them are just H's. So that is our beta glucose, and how these would join together. Well, we've got these OH groups there. This would, we've got another molecule there, then these would react in a different colour. We would remove water, H2O, and that would leave us with, just join the sticks up, the bonds, that would leave us looking like that, and the name of that would be our glycosidic bond. So we just need to put that into words. We are forming a glycosidic bond and that's between carbons 1 and 4. So it's our carbon chain, so 1, 2, 3, 4, 5, 6, and this is the first carbon of the next molecule on. So it's a 1,4 glycosidic bond between carbon 1 and 4. Or you might have said a 1,4 glycosidic bond, that would have got that mark as well. And we only need 2, so if you said a 1,4 glycosidic bond, then that would have got you 2 marks. We've mentioned this condensation reaction. So condensation reaction, removing water. So 4 things you can say, and you only need 2 of them. And that's our, I've written that in the wrong place, sorry. Right, what do you do if you write it in the wrong place? It's because I drew this diagram down there. I am going to cross it out completely and write it here. So I'm going to write glycosidic bond between carbon 1 and 4, condensation reaction, removing water. So now that's right. I'm going to just put a line through this. I'm not going to draw any silly arrows, that will confuse the examiner. And now I'm going to write in the right answer here. So Explain how the structure of cellulose makes it suitable for use in plant cell walls. Well, when we get these long chains, we have glucose monomer units, but they actually, as you probably know, one faces up, one faces down, because these bonds don't like to go across. It's not very stable for them to go across. They tend to form on one side or the other, and you tend to get the glycosidic bonds forming like that. But that makes long straight chains, and these actually get hydrogen bonds between them and form kind of bunches of microfibrils, which makes them very strong. So here we can say long parallel chains hydrogen bonds between chains and microfibrils formed, and these are strong. So plant cell walls are very strong, these cellulose cell walls, and that's why plants don't fall over, because of these microfibrils formed from long chains of cellulose with interconnections with hydrogen bonds. So really, for 14 marks, that should have been fairly straightforward. A lot of AO1 knowledge that you were just repeating onto the paper.